Let's go ahead and take a step back and focus on the big picture of glycolysis for a second. Up till now, we focused on the very first step of glycolysis, which involves taking a glucose molecule, using our hexokinase or glucokinase enzyme to phosphorylate it, making our glucose 6-phosphate. Now, we're going to see glycolysis substrates, intermediates, and end product. I'll first focus on the important steps, but I wanted to first show this large picture of glycolysis to you. Notice how most of these steps have double-headed arrows, meaning they can occur in both the forward and backward reaction direction. Here, we're going to see 3 steps of glycolysis that are irreversible, meaning they can only occur in the forward direction. We also have our net reaction for glycolysis seen by this reaction here. What we're going to do is we're going to start off with a molecule of glucose, 2 inorganic phosphates, and 2 ADP, as well as 2 NAD plus electron acceptors. We've already talked about those. And what we're going to do is we're going to undergo glycolysis, and we'll end up with 2 molecules of pyruvate, 2 molecules of ATP, 2 NADHs, hydrogen, and water. Now, what is the important takeaway message for this slide? Well, the important part to see is that you need 1 glucose molecule to make 2 pyruvates and 2 ATP, as well as, in the process, 2 NADH molecules get reduced into our NADH molecules. Now let's go through the enzymatic steps that is going to regulate glycolysis. The 2 steps in glycolysis that require ATP are going to be our first step and our third step in the unilateral direction. Notice how it has a star next to it. This is going to represent our initial energy investment phase. So first, we've already said that hexokinase or glucokinase is going to convert glucose using 1 ATP into glucose 6-phosphate. Now, as I mentioned before, our glucose 6-phosphate is going to act and inhibit the hexokinase enzyme, but it's not going to affect our glucokinase enzyme. Instead, high levels of fructose 6-phosphate are going to act to inhibit our glucokinase. In the third step of glycolysis, phosphofructokinase 1 is going to use 1 ATP to convert fructose 6-phosphate into a fructose 1,6-bisphosphate. This just means that it's still a fructose molecule that has a phosphate at the 1 and the 6 position. So, do you remember what the rate-limiting enzyme was for our glycolysis pathway? Remember that chart earlier on in the biochemistry metabolism section where it had a large chart and we saw all the rate-limiting enzymes? Well, our phosphofructokinase 1 enzyme is going to be our rate-limiting enzyme in glycolysis. So there are a lot of things that either activate it or inhibit it. For example, if a cell has a lot of adenosine monophosphate, or AMP, this must mean that they have to make more ATP, so PFK will be activated. And if there's a lot of fructose 2,6-bisphosphate around, this indicates that the blood sugar is high, so there's plenty of glucose to be used for more glycolysis, so both of these are going to activate our phosphofructokinase 1 enzyme, thus speeding up glycolysis. If lots of ATP and citrate are present, this means that the cell has a sufficient amount of energy available and it does not need to continue on with glycolysis. Remember, glycolysis is breaking down a glucose molecule to make more ATP, hopefully. There are 2 steps in glycolysis that are going to produce ATP as well. But both of these occur twice, since they occur after the step where our 6-carbon molecule is split into 2 3-carbon molecules. So notice how we have a 6-carbon molecule here. It gets broken down into 2 3-carbon molecules. So this means that a total of 2 ATP are initially consumed. Here's 1 and here's 2. So these are 2 total ATP. However, we're going to get 2 ATP out in our energy out phase. So we can see that here, and we get it twice since there's 2 3-carbon molecules. So if we do some math, we'll see that we get some excess or some profit of ATP. We have already invested some ATP prepping this glucose molecule. So what steps do we finally make the ATP? Well, the first step of glycolysis that produces ATP uses the enzyme phosphoglycerate kinase. Phosphoglycerate kinase is going to convert a 1,3-bisphosphoglycerate molecule into a 3-phosphoglycerate molecule. Going by the names of these, it makes sense that 1 ATP was produced since 1,3-bisphosphoglycerate has 2 phosphate groups, which are at the 1 and 3 position, while 3-phosphoglycerate only has 1 phosphate group, and that's at the 3-carbon position. So that means that one of those phosphate molecules was taken off and in the process added to our ADP to produce one molecule of ATP. Why does this enzyme, phosphoglycerate kinase, work twice as hard as, let's say, hexokinase or phosphofructokinase 1? This enzyme has to work twice as hard because, remember, halfway through glycolysis, we take a 6-carbon glucose molecule equivalent, or fructose, and split it into two 3-carbon molecules. And these two smaller fragments, called glyceraldehyde 3-phosphate and DHAP, which gets converted into a glyceraldehyde 3-phosphate, these are going to undergo the same reaction to form two molecules of pyruvate. And then finally, we have another step that's going to produce ATP. This one is going to use the enzyme pyruvate kinase. This is going to convert phosphoenolpyruvate into our pyruvate. So this is activated by fructose 1,6-bisphosphate, which is an intermediate of glycolysis. So remember, fructose 1,6-bisphosphate is going to be right there, and this is going to activate our pyruvate kinase. Pyruvate kinase is thus inhibited by high levels of ATP and also alanine. Since again, if you already have a lot of ATP present, you don't want to keep on going through glycolysis just to make some more. You already have enough ATP. Again, pyruvate kinase is going to work double time in glycolysis, just like we saw before with phosphoglycerate kinase, because we have two 3-carbon molecules that it has to go through. And that's it for the glycolysis pathway. So we'll now need to transition to another metabolic process, the TCA cycle. Pyruvate dehydrogenase, PDH, pyruvate dehydrogenase, is the enzyme that's going to link our glycolysis, which we just talked about, which takes place, remember, in the cytoplasm, and our TCA cycle, which is going to take place in the mitochondria. So we have an enzyme complex, the pyruvate dehydrogenase complex, is going to transition us from pyruvate, and we're going to take us into our TCA cycle. So how well were you paying attention? In glycolysis, what reactions require an investment of ATP? Well, there are two steps in glycolysis that actually require an investment of ATP, converting glucose to glucose 6-phosphate and also fructose 6-phosphate into a fructose 1,6-bisphosphate. It's like investing on Wall Street. You need to initially invest some money, so 2 ATP in this example, before you get a return on your investment, or 4 ATP, and also some NADH molecules. We already took a look at glycolysis and in part of how it was regulated. It's pretty logical to understand why end products such as ATP can inhibit glycolysis. But it might not be as intuitive for the reason why fructose 2,6-bisphosphate is going to activate glycolysis. So how does fructose 2,6-bisphosphate fit into our nice glycolysis pathway? So notice how I took the first 3 steps of glycolysis and kind of made them bigger. So how does fructose 2,6-bisphosphate fit in? Well, I like to think of fructose 2,6-bisphosphate as a regulation molecule, or a side process that's going to go alongside of glycolysis. It gets made from a phosphofructokinase 2 enzyme, which is just a cousin to the phosphofructokinase 1, and our fructose 2,6-bisphosphate gets broken down by fructose bisphosphatase 2 enzyme. Our fructose bisphosphatase 2 enzyme and our phosphofructokinase 2 enzyme are actually part of the same protein complex, but it responds in opposite ways to phosphorylation by the protein kinase A. So this is actually part of just one big complex, and it just depends on whether it goes in the forward direction or the backward direction based on whether it's phosphorylated or not. So let's compare the regulation of this enzyme in the fed and fasting state. So let's go ahead and say in the fasting state, what is going to happen? Well, in the fasting state, high glucagon levels is going to increase the amount of cyclic AMP through the G protein coupled receptor system. This is going to actually increase the amount of protein kinase A, and like I said earlier, protein kinase A is going to phosphorylate our fructose bisphosphatase 2. So we're going to add a phosphate group onto it. This phosphate group is actually going to activate our fructose bisphosphatase 2 enzyme, and it's going to inhibit our phosphofructokinase 2 enzyme. So it'll work in this direction. This means that you're going to convert this side molecule of fructose 2,6-bisphosphate into molecules of fructose 6-phosphate. And you're actually going to inhibit this phosphofructokinase 2 enzyme, so you're going to inhibit this reaction here. This is going to allow gluconeogenesis to occur and replenish blood glucose levels, because if you're in the fasting state, you'll want to undergo gluconeogenesis. You'll want to create new glucose molecules, so you'll want to stop glycolysis and you'll want to activate gluconeogenesis. It makes sense if you're in the fasting state. So let's go ahead and clear all this off. Now, what do you predict will happen in the fed state? So if you have a large meal, for example, well, in the fed state, high insulin levels are going to decrease the amount of cyclic AMP, which is going to therefore decrease protein kinase A activity. Now, the enzyme fructose bisphosphatase 2 will be dephosphorylated. Remember there was that phosphate group attached? What this is going to do is we're going to dephosphorylate it, meaning only the phosphofructokinase 2 enzyme is going to be active. So this will be activated now, and this will be crossed out. What this will do is the body will start converting our fructose 6-phosphate into molecules of fructose 2,6-bisphosphate, which is going to activate the forward reaction by phosphofructokinase 1. So it's going to actually activate our phosphofructokinase 1, which we already talked about earlier. Just remember, the molecule fructose 2,6-bisphosphate is made to regulate the steps of glycolysis. Keep in mind that phosphofructokinase 1 is still the rate-limiting enzyme step of glycolysis, and therefore it's important to regulate this enzyme more than any others in glycolysis. So here we have this little side reaction going on. Depends on whether it's phosphorylated or not. Depends on if it's in the fed or the fasting state. But remember, fructose 2,6-bisphosphate, the whole concept of this is to regulate a step of glycolysis, and that step is our rate-limiting enzyme step. So let's do a flash quiz. A hard-working medical student on her surgery clerkship is going to skip her lunch to go to the operating room. She is a gunner. How does this affect the action of fructose bisphosphatase 2 and the phosphofructokinase 2 enzymes? So she's fasting. In the fasting state, increased glucagon levels are going to increase cyclic AMP levels and also activate our protein kinase A activity, which is going to phosphorylate our fructose bisphosphatase 2 enzyme, ultimately activating it. So it's going to activate this enzyme and also at the same time inhibit the phosphofructokinase 2 direction. So we're going to increase our gluconeogenesis, and at the same time we're going to decrease our glycolysis in the fasting state.